Good evening, everybody. So we're very pleased to be partnering with the St. Louis Zoo to bring you tonight's science seminar. And for those of you who aren't familiar with the Academy of Science, I'm going to take just a couple minutes to tell you a little bit about who we are. My name is Rose Jansen, and I'm with the Academy. We are an independent science organization. We are supported entirely through community contributions. And at venues throughout the region, we connect science in the community through free and our very low-cost public talks, science seminars, and trips and tours that celebrate science at venues throughout the metropolitan region. We also offer a full array of free and low-cost opportunities that are expressly for teens, such as our Teen Science Cafes, Youth Leadership Council, Junior Academy of Science, and the Regional Academy of Science St. Louis Science Fair and its Honors Division. The Junior Academy is a pre-professional STEM membership organization for students from the full range of academic backgrounds in grades 6 through 12. It offers hands-on opportunities in science, engineering, and medicine. Teens can attend unique behind-the-scenes explorations of leading engineering, technology, and science labs of our region, access university libraries, participate in field opportunities, and challenging and engaging science competitions. Junior Academy members make real-world connections, meeting top STEM professionals. You can find more information on the Junior Academy and all of our community-wide science events and programs by visiting our website at academyofsciencestl.org. You may also visit us on Facebook, Instagram, or Twitter, or before you leave tonight, pick up some of the literature that is on the table just outside the auditorium, or you may sign up to receive Academy e-news. There will be some e-news sign-up sheets that will make their way around the audience tonight. I do want to mention a few upcoming Academy events you might have an interest in attending, and if you are a student, you might want to check with your teacher. Many teachers offer extra credit for attending Academy events, and we do provide proof of attendance if needed, and you can get that from us following tonight's Q&A at the table outside the auditorium. Tomorrow night at 7 p.m., Washington University Professor of Environmental Engineering Science, Dr. Richard Axelbaum, will be at the Bridgeton Trails County Library to talk about combustion experiments aboard the International Space Station. This event is free and open to the public, but you do need to register to attend, and you can do so by logging onto our website at academyofsciencestl.org. If you are a teen or know a teen who is interested in hands-on opportunities in science, next Monday from 5:30 to 7:30 p.m. on the Washington University in St. Louis Danforth campus, teens in grades 6 through 12 are welcome to join us at our Teen Science Cafe on cell biology, how cells get in shape, with biology professor Dr. Ram Dixit. This event is also free, but you do need to register to attend, and again, you can do so by logging onto our website, and we do serve dinner. On Tuesday, March 13, from 5:30 to 7:30 p.m. at the Sheet Metal Workers Local 36 on Shoto, you are welcome to join us to explore the mission and metrics behind Paul Hawken's Project Drawdown, the most comprehensive plan ever proposed to reverse global warming, including practical local solutions in action. Left Bank Books will be at the event with copies of Project Drawdown for purchase. This event is free and open to high school students and adults. You can register to attend on our website. And then again, if you are a teen, you might have an interest in joining us on the Webster University campus on Thursday, March 15 from 9 a.m. to 2 p.m. for an interactive and hands-on science careers exploration of the STEM and tech in media and communications careers. Interactive and hands-on sessions explore exciting fields of study such as animation, audio engineering, game design, radio broadcasting, and visual processing. This event is also free and open to students in grades 6 through 12. You do need to register to attend. And then lastly, if you are a student and you need to verify your attendance at tonight's talk, as I mentioned earlier, we will have verification cards available following tonight's Q&A at the table outside the auditorium. Please turn off cell phone ringers or any other electronic devices that might make noise during tonight's program. With that said, I'd like to introduce tonight's speaker, Dr. David Van Essen. Dr. Van Essen earned his undergraduate degree in chemistry from the California Institute of Technology and his Ph.D. in neurobiology from Harvard Medical School, where he also completed a postdoc in neurobiology and then went on to earn a second postdoc in neurobiology at the University of Oslo, Norway. Dr. Van Essen is an Academy of Science St. Louis Fellow and the Academy's 2007 Peter H. Raven Outstanding St. Louis Scientist Lifetime Achievement Award recipient. He is currently an Alumni Endowed Professor of Neuroscience with the Department of Neuroscience at Washington University in St. Louis, where his research focuses on the structure, function, connectivity, development, and evolution of the cerebral cortex in humans and non-human primates, and his lab develops and uses computerized brain mapping techniques as well as neuroinformatics tools that facilitate data analysis and data mining. He is heavily engaged in and has served as the principal investigator for the Human Connectome Project. He'll tell you a little bit about that tonight. It's a large-scale collaborative project to map human brain circuitry in healthy adults and that circuitry's relationship to behavior. And he is here with us tonight to talk about his work mapping the human cerebral cortex. On behalf of the Academy of Science St. Louis and the St. Louis Zoo, won't you please join me in welcoming Dr. David Van Essen.
Thank you for that kind introduction. I'm delighted to be here to talk to you about my favorite scientific subject, the human cerebral cortex, and perhaps I need to do something to get us a full view here. There we go. So, all aboard. Our quest tonight is to learn something about how the human brain works, to make us unique as a species, unique as individuals. Everyone in this room is different. We're gifted with many amazingly diverse talents. We're afflicted, all too many of us and our friends and family are afflicted by devastating brain disorders at all stages of the lifespan. So I would say that deciphering the brain in health and disease is really a grand challenge for the 21st century and beyond. So let's take a closer dive into the brain. Here's another view of the brain from what we call the side, the lateral side. And if we slice the brain right down the middle, the midline, and take a peek from what we call a medial view, you see some more stuff lurking. And if we take a slice in the what we call the coronal direction or orientation, you see what's shown on the right here. Things are gray and white, and lo and behold, we call this gray matter and we call this white matter. And this outer wrapping that's very crinkled, we call the cerebral cortex. Now you see it from several views. And the cerebral cortex is the dominant structure of our brain in terms of brain weight. Our brains weigh almost 3 pounds, and 80% of it is cerebral cortex. And it subserves a tremendous range of functions that we'll learn more about as we go along. Remarkably, the cerebral cortex contains only 20% of the neurons in your entire brain. That's because it's chock-a-block full in this gray matter with trillions and trillions of synapses, the connections that communicate from one cell to another. And underneath the gray matter is the white matter I mentioned before, that is the cables that connect one region of gray matter with many others all around that we'll learn more about. So altogether, in this white matter, if you add up every single nerve fiber, its length, you end up with an estimate of about 100,000 miles worth of axons or fibers within the white matter. This tiny little structure off to the left on this panel over here is a pint-sized structure we call the cerebellum. It's only 10% of the total brain mass, and yet it contains about 80% of all the neurons. And it is involved in coordination of many things that we do, our movements, our thoughts, and many of our actions. I'm not going to say much more about the cerebellum. It's very important. Another part that I'll just touch upon briefly is this collection of structures that we call the rest of the brain. It's less than 10% of brain mass. It has less than 1% of all the neurons. We call them a collection of subcortical nuclei, little clusters or clumps of gray matter that are very different from the cerebral cortex. These subcortical structures are in aggregate extraordinarily important for keeping us alive and well, making us breathe, making our heart beat at the right pace, making us regulate our hunger, our sleep. Fascinating, but I don't have time to talk so much about them. But our overarching challenge is to untangle the connections of the brain, because that's what makes the brain work, the communication between nerve cells locally and at long distances. So the way we're going to go about business is to start with some brain basics, consider some analogies with computers on the one hand and genomes or genetic code on another. I'm going to interject a little bit more of the personal background to tell you how and why I became a scientist, and then we'll jump back into brain mapping with what we call cortical cartography, as well as the wiring and the connectivity. I'll revisit this issue of variability because that's fascinating and very fundamental, and we can get some insights into differences from one brain to another. I'll show you examples of something that's a very cool mapping technique. We call them Myelin Maps, and I'll say why. And we'll have our introduction to brain function using a classical technique of positron emission tomography, but we'll spend most of our time on the Human Connectome Project and the advances that it's brought us in understanding brain structure, function, and connections. So here's that brain again, and here over on the right is a computer. This in 1997 was the world's most powerful computer. It sure isn't anymore. But let's do some comparisons anyway. So inside the brain, if you look under the microscope at one small region, you see it's containing many cells, neurons, nerve cells we call them. And the cell body, where the housekeeping machinery is that makes proteins and keeps the cell alive and well, is this little triangular patch right in the center, or this little round patch over for another cell here. What makes nerve cells fascinatingly different and far more complex than the cells in your liver, your stomach, your lungs, and so on, is the appendages that neurons have. So they have a collection of branches that you see I'm pointing to here. These are called dendrites, and they're the receiving ends. Think of it as a little tiny radio antenna that picks up signals from other sources. And it listens to those signals and decides whether to send a message, a nerve impulse, down the single solitary branch that we call an axon that goes long distances and then branches to make connections with target cells. And those connections, as I mentioned before, we call specialized communication units called synapses. Very fancy machinery, and we've got a lot of them. And each neuron has, on average, about 10,000 of these synapses or synaptic inputs on them. And each of these axons that communicate over long distances carry nerve impulses or action potentials, as I said, that are actually rather slow in their how rapidly they're conducted and how many impulses per second, up to 100 per second, which might seem like a lot, but it's actually nothing compared to what goes on in computers. So to return to our computer analogy, this deep blue computer was the world's chess champion in 1997, and it had inside its computing interior little computer chips, which are basically specialized transistors and fine wires that can literally carry out billions of calculations per second. And that was then. Nowadays, computers are vastly thousands or if not millions of times more powerful and more rapid in their computations. They're getting more and more powerful as we speak. We still like to think we're smarter than computers. Maybe they'll tell us someday that, no, we got it wrong. But let's keep up with that illusion, at least for the time being, and try to consider why and how we think and compute and how it's different from computers, even though there are some first-order analogies. So we think of this as the connectomes are the complete set of wires that let us do our daily tasks. And if we drill down farther, the next stage of our comparison is not with computers, but with the genome. So many of you know about the genome, the Human Genome Project you perhaps have heard about. And that was an enterprise that was centered at Washington University, among other places, and discovered that each person has a genome, a set of 3+ billion base pairs that are the genetic code that were sequenced and now can be routinely sequenced with incredible accuracy. But let's take some basic comparisons. We have about 3 billion base pairs in our human genome. A little mouse has almost as many base pairs in its genome. And if we look at the genes, the instruction kit that makes proteins and other features that are the daily life of cells, we have in this whole genome an estimated little more than 20,000 genes, and the mouse has about as many, perhaps a few more. So what makes us so much better and smarter? Well, our brains are vastly bigger. So here's a mouse brain, and to scale a human brain, and there are thousands of times different in size. And if we look at the number of neurons, I'd already mentioned there are almost 100 billion neurons in your brain, and there are much fewer, only 70 million or so neurons in a mouse brain. If we look at the parts of the brain that I've already alluded to, they're kind of equivalent to the gene as a fundamental unit or part of the human genome. So the structures that make up the anatomical parts of the brain include various nuclei underneath the cortex, the subcortical nuclei, and we have hundreds of them, mice have hundreds of them. There's not a dramatic difference in that. And if you look at the cerebral cortex, it's a mosaic of many different areas. I'm going to return to that in detail, but just for a first pass, let me note that there are only about 40 cortical areas, functioning units of the mouse, and the question of how many cortical areas the humans have has been debated for more than a century, and the answers have ranged from as few as 50 to as many as 200. I'm going to revisit that and vote for the larger number, but we'll have to take a little while to get to that part of the story. So I told you I'd give you a little more background. When I was in high school and even younger, I really wanted to be a scientist, and I thought I wanted to be a physicist, and that sounded very cool. I went to undergraduate school at Caltech, and I decided physics was actually too hard for my poor brain, so I'd choose something that was a little bit more manageable, and I decided to become a chemist. Then literally I was in a bookstore one day and picked up this book, and it was about the brain, called The Machinery of the Brain. I read it, and I've become a neuroscientist, hooked solidly ever since. That took me to Harvard for graduate school, to a newly formed Department of Neurobiology. At that time, neuroscience wasn't an in thing. It was an aspiring young field, but I became a graduate student and studied a very simple nervous system. I didn't want to tackle something as complex as the human brain. It took me decades to move in that direction. But as Rose mentioned, I took a journey as a postdoctoral fellow to Europe, England, and Norway. That was great. And I learned to study not only leeches, but I jumped into the visual cortex and also how the brain develops. Then I returned to my roots at Caltech in the mid-'70s and continued research there. And then I was lured 26 years ago to WashU, where I've been ever since, and that's gotten me into the Human Connectome Project. And you'll learn more about what I'm currently heavily engaged in, the so-called Lifespan Development and Aging Project. We'll come back to that. Let's focus now on brains inside the skull. So here's another picture of the human brain, a little crisper and cleaner. And I already mentioned we're thousands of times larger than a mouse brain. We're several times larger than our nearest living relative, the chimpanzee, more than tenfold larger than the macaque monkey, an intensively studied laboratory animal that we'll talk about a lot. For good measure, here's a small-brained primate, the marmoset, and we're 200 times larger than that. So there's tremendous diversity in brain size. And the other thing you can see is that the folding patterns are dramatically different. The mouse is very smooth. The marmoset has a smooth cortex. Monkeys and apes have much more folded or convoluted cortex, and ours are staggeringly complex. So let's revisit that. We can see those convolutions or folds in a magnetic resonance imaging device, an MRI. How many of you have actually been inside an MRI scanner? Lots of people. So you've probably seen images like this. This is the structure of white matter and gray matter. And this is not a physics lecture, but I just want to give you a little bit of orientation to what goes on in the scanner. When you put your body on the table and your head in the scanner, you're putting inside it a very powerful magnet that has a magnetic field far greater than that of the Earth. That's the outer big superconducting magnet that's on the exterior. Inside that is a ring of what are called electrical coils that generate changes in the magnetic field. Static magnetic fields doesn't get you much information. You need to rapidly change the magnetic field, and you also need to probe how the brain is responding to those changes in the magnetic field with an electrical measurement that's called a radio frequency coil that generates things that are called pulse sequences. These are what make the noise, the sounds that when you're in a scanner, the humming and bumping and grinding. And they're all different depending on what kinds of information you want to get. And then to see what's going on, one uses something that's called a head coil. That's what's literally around your head inside the scanner, and that detects extraordinarily faint signals and sends those to computers, which are able to decode the signals and reconstruct the images and allow for some very fancy analyses that we'll see. So here's another movie, decades old, so kind of a grainy movie going from the back to the front of the cerebral cortex above and the cerebellum below, and the little black patches there are the fluid-filled ventricles that are in everybody's brain. So it gives you a sense of the complexity of the cerebral and cerebellar cortex. And if we take a brain, what you just saw was from a living brain. If you take a postmortem brain after death and slice it with a knife and cut very thin slices and then stain those in various ways, one way of staining the brain tissue shows where the cell bodies are. So the gray matter now is turned purple because that's where the dye adheres to in the cells. And if you magnify one region of that, you can see actually individual nerve cells where their cell bodies are, and you see in some layers they're packed densely, and other layers they're packed sparsely, and sometimes from one region to another, the appearance or architecture changes, and we call that a boundary from one functional region to another. And that's reflecting the organization of these cells and their connections. I mentioned that each neuron has an axon that goes long distances. Let me just finish the cytoarchitecture story. A famous anatomist a century ago, Brodmann, made a map of one individual postmortem brain, and he was the one who said there are about 50 areas using that kind of mapping method. Another set of investigators took advantage of the fact that axons are insulated by a fatty wrapping that makes signals conduct faster, and that was called myelin. And if you stain another section of the brain for where there's myelin, you see, as you'd expect from what I told you earlier, that the white matter is now turned jet black because it's packed full of these tiny axons. But within the gray matter ribbon, there's some degree of myelin that differs from one region to another, and we can see that in a microscope, and we can look for those changes to see whether they give evidence for regions that differ from one another in their appearance, and the hypothesis is that also tells us something about function. So the classical anatomists, the Cecil and Otto Vogt, also in Germany, proposed from their analysis that there were upwards of 200 distinct cortical areas. So this is what set up this essentially a dynamic tension that the field has wrestled with for a full century. So to understand what's going on, it's not entirely satisfactory to just show maps on the convoluted brain without recognizing the fact that it's very folded. And that's something that got me intensely engaged when I started working on monkey visual cortex. And I use the analogy of flat maps of the Earth. So here's a book atlas that shows the Earth with cuts that let you flatten out the spherical Earth. And we use it, for example, to display geography where there are hills, valleys on the land, or deep trenches in the underlying oceans. And I did the same thing for the macaque monkey. So this is a flat map of the entire right hemisphere, cerebral hemisphere of one macaque monkey. And this map is showing what we call a geographic map The shaded regions are the regions that are buried in one or another of these folds that we call sulci. By the way, if I'm unclear along the way and you have a question for clarification, I'm happy to stop. Please raise your hand and I'll answer it. If you have questions that you want to dig deeper in, we can return to that at the end. Okay, so here's the same monkey flat map now, painted, colored, to show a map of cortical areas. The ones that are colored are associated specifically with vision, the sense of sight, and monkeys like us are intensely visual creatures, so there's a lot of cerebral cortex devoted to processing that information and seeing what is it out there. Is it a face, a shoe, or whatever? And so this mosaic of dozens of distinct areas associated with vision are identified using a combination of methods, looking for differences from one region or another in function, in ways we'll come to, in architecture that I've already shown about, in connectivity, where the wires are going. There are fancy ways to trace connections, and in what we call topography, recognizing that each cortical area that's associated with vision has a map of the external world. The three-dimensional world gets focused on the eyes, and it gets mapped into a representation that's an orderly mapping in each visual area. So some visual areas in this early 1990s summary map of the monkey are well defined by any and all of these distinct approaches or ways of characterizing cortex. So here, this big one is the primary visual cortex. Here's a little one we're going to visit again. It's called MT. That stands for the middle temporal area, but remember it because it's involved particularly in processing motion, things that are moving around in the visual world you care a lot about, and part of that analysis occurs in this little area. But most of these other areas, although they have crisp boundaries on this summary map, the data that scientists, we and others used, was not so clean. The boundaries were fuzzy, and they weren't the same from one study to another. And so this was one version, but it's evolved, and not everyone was sold on that particular summary map. So the reason for mentioning that is that this parcellation problem that we've visited several times is a profoundly difficult one, but there's dramatic progress that we'll come to from work from the Human Connectome Project. But we have a little ways to go before we get there, because I want to talk about individual variability in development. So here's another movie through a structural MRI volume. It's crisper and cleaner because it's higher quality data with higher spatial resolution for imaging each slice. And the surface model here, the 3D model that you see on the right, is generated from what we call an algorithm, a computer algorithm that captures the shape of the cortical folds and represents them very accurately. And one of the beauties of having this 3D model is that we can manipulate its shape. You've seen it just inflated, smoothed out so we can see inside, and now the darker it is on the streaks of the inflated surface, the deeper it was in one or another part of the 3D model. Does that make sense? Okay, so now here's a top-down view of the left and right hemisphere from one individual brain. We can not only inflate these surfaces, we can flatten them out computationally, just as I did with an old pencil and tracing paper method for the monkey cortex earlier. But now I let you find a useful analogy. Each hemisphere inside your skull is about the size of a 13-inch pizza, and the cortical areas we're going to say a lot more about, think of them as kind of pepperoni-sized little areas, but some are bigger than others. We'll return to that. But now this issue of individual variability. So each of you has a highly convoluted cortex that looks qualitatively like these 4 healthy young adults that are scanned here on the left panel. And the wrinkles don't look exactly identical, but it's hard to tell what's different from one person to another until you inflate them, as we've already seen in the movie, and see the folding patterns mapped on this smoothed out cortex on the right. So here are these same 4 individuals. Now the patterns don't look the same if we look in detail. In some regions, like the one at the very top here, they're very similar. Can be seen, some folds can be seen in every individual. In other individuals, there are very different folding patterns from one to another. Remarkably, these 4 individuals were not taken randomly off the street. They're a pair of healthy young adult twins. And I won't play the guessing game with you. I'll just tell you, down here is one pair of identical twins. Up on top is another pair. And look, even though these twins have the same genetic instruction kit that said grow a brain, the folding patterns are strikingly different from one person to the next. So that tells us that there's tremendous variability in twins. But if you look closely, as we did years ago, there is some heritability, but it's not what hits you at first glance. Another important thing is these cortical areas that I've said something about are variable in size. So here are, on the bottom, are 3 brains of different individuals, and the red shows the primary visual cortex, like you saw in the monkey. Now this is in a human brain. And the position of these primary visual areas are not exactly the same from one person to the next, and they even vary by 2-fold from one person to the next. So when you think about how you're better at one thing or another, it's intriguing to speculate that that's because you may have more cortical tissue devoted to one type of processing versus another. That's still a mystery, but it's one we're fascinated by. But these examples here bring us to another question, which is, what makes the cortex fold anyway? Why is it crumpled up inside the skull? And that question becomes even more intriguing when we think of not just the few primate examples I showed you before, but think of a dolphin, an elephant, a camel, and then the very smooth-brained manatee. So there's tremendous diversity across the mammalian realm. And the cerebellum, I'll just mention in passing, is even more convoluted and has a different folding pattern. But let's focus on cerebral cortex and think about what's going on. So we know that when folding occurs is late in pregnancy. So during the 3rd trimester, the brain goes from a very smooth shape with just major deep folds to a highly convoluted pattern. And in work that Donna Durker and others did with us at WashU, we segmented these MRI slices of individual brains at different ages, and we could see that over the 3rd trimester, the brain and the cerebral cortex becomes progressively more complex. And if we cut to the chase here, between the beginning and the end of the 3rd trimester, a tremendous amount of folding occurs, and at term equivalent, when a child is born, the cerebral cortex is nearly as folded or convoluted as it is in the adult, even though it's only 1/3 of the size. But again, what makes it fold? Well, I had my favorite scientific light bulb moment was when I was musing on some findings that Tom Coogan and I had published in years ago, when we looked back at a monkey cerebral cortex, and we showed that V1, the primary visual cortex, and the neighboring area, a large one called area V2, become wired, become connected together in both directions right around the time that a fold forms in between them, that goes from the smooth to the folded. And the idea then is that maybe these connections are not just coincidentally correlated with folding, but they may be the cause. And the idea, more specifically, is that mechanical tension, think of each axon generating a little tiny tug between where its cell body is and where its termination is. And if everybody pulls together, a little bit of force working from large numbers of axons will bring strongly connected regions closer together. And that's by this idea of what forms a gyrus. And then the opposite kind of fold, what we call a sulcus, is essentially the outcome of a losing battle. There wasn't enough force to fight against that. So this simple hypothesis is not proven, but it can account for the folding that's consistent in some region, connecting strongly connected regions, and variable in regions that are a little mosaics of what we think of as balkanized parts of cortex. So it's probably not the only mechanism. There are also other developmental influences, but it's at least a framework for thinking of how can something so marvelous and mysterious actually be explained by simple developmental principles. So I'm now going to turn back to the adult brain, and first of all remind you of a slide that you saw before. One way of staining shows you where the cells are. That gives us what we call cytoarchitecture. Another type of staining, post-mortem, after death, gives us maps of where the myelin is in the white matter and in the variable degree of myelin in the gray matter itself. So now we're going to go back from the post-mortem to a living brain, and we're going to take a slice through a living brain now horizontally, not vertically as you see here, and this is the image you see over here. So when my very talented student Matt Glasser came to my lab, he had previously noticed that in a standard brain image, like you've seen already in the movies, some regions of the gray matter ribbon are a little bit brighter than average, and other regions are a little darker than average. One might have said, it's just noise in the data, but Matt noticed that in a different kind of brain image acquired in the same time frame, but a different pulse sequence, as I alluded to, this is called the T2 weighted image, and it's obtained from the same individual, but now the white matter looks dark, the outer cerebral spinal fluid looks bright, and the gray matter still looks gray, but the regions that had been brighter were now darker, and the regions that had been darker are now brighter. So taking the simple mathematical ratio of one value versus another and coloring it gave us what we call maps of myelin content. And we call it Myelin Maps because in mapping this data to the cortical surface, we see that the red regions that have a high ratio are the regions that we know from postmortem histology are heavily myelinated, and these much larger blue and indigo regions are lightly myelinated. And we can see this not only in human brains, but also in our animal models. The chimpanzee has a similar pattern, the macaque monkey has a similar pattern, and we can see distinct cortical areas in each species, even though the sizes are different. And we can see that up here in the frontal lobes and in the temporal lobe and in another region out on the side, it's all lightly myelinated, but we have a lot more of that, and that's important because these lightly myelinated regions are the ones that are associated with higher thought processes, cognitive processes. So in essence, the hypothesis is we're smarter because we not only have bigger brains, we have more devoted towards these higher cognitive functions. So let's now move on to brain function. And positron emission tomography was invented decades ago, and Washington University was a leading institution for using it to map brain function. So here's a study done by Mark Rakel, where looking at inside the PET scanner, as we call it, if instead of looking at a blank screen, one looks at a checkerboard that's flashing on and off, that's a strong visual stimulus, and it turns on regions at the back of the brain that are associated with vision. So the difference between stimulating and a baseline or so-called control is a strong activation of visual cortex. And working with Steve Peterson, Mark Rakel, and others showed that there are specialized regions not only for vision, but for language. So reading nouns, generating verbs, cognitive processes now are shown to activate different brain regions, but only in a coarse and fuzzy way because the methods didn't have the resolution that we now have. But this gave us enough to show that, like the monkey brain I showed you before, we can make a map of functional organization of visual cortex, movement or motor cortex, tactile or somatosensory cortex in different brain regions, and then in yellow, these large regions devoted to thinking or cognition. Okay, now let's finally move on to the modern episode of looking at the brain through the lens of the Human Connectome Project. I'll tell you what the idea was and what is accomplished so far. So I touched upon the word connectome before, but let's just revisit it briefly. It's a comprehensive map of neuronal connections. It aims to be comprehensive, just like the genome, as you may know, is the full sequence of all base pairs with incredible accuracy. But for the brain, our methods don't let us get complete accuracy and complete coverage. So there is a world of using electron microscopy and looking at the exact location and identity of every nerve cell, axon, dendrite, synapse, and coloring it and labeling it. But it can only work currently for extremely tiny bits of tissue. There's another world using animal models to use pathway tracing methods and trace connections long distances with moderate resolution, but not at the same as the electron microscope. We're living tonight mainly in the world of the macro connectome, as I like to call it, looking at the whole brain and long distance connections and interactions, but only with limited resolution. Each of the little volume elements that picks up signal is a millimeter or two on a side, and that's literally containing hundreds of thousands of neurons and trillions of synapses. So what is the Human Connectome all about? It started 8 years ago in 2010, went for 6 years, and the objective was to get comprehensive data on brain structure, function, and connectivity in healthy young adults, twins and their siblings, using better MRI scanners, better adaptations of these things I call pull sequences, and get 4 hours, an unprecedented amount of data from each healthy young adult volunteer. And the data was, in fact, exceptionally good. We studied almost 1,200 subjects, and we got a huge amount of behavioral and what we call demographic data from each subject, hundreds of different measures, and we got genotyping information. We've analyzed the data using improved methods that you'll see examples of. And then we've shared this data freely and publicly to the scientific community over many years. And more than 10,000 investigators have bought into this. Well, they didn't pay, but they've tapped into our data sets. They've gotten more than 16 petabytes, that's 16,000, so I'll typo there, terabytes of data, and hundreds of publications to date have emerged from using this data. So what does the data look like? Why are we excited about it? Well, Deanna Barch, who's in the psychology department at WashU, spearheaded one approach, which was what we call task fMRI, having subjects lie in the scanner and do something. And they did, over a period of an hour's worth of acquisition, 7 different tasks. One was just listen to stories and answer questions. Another is do arithmetic and get the right answer. Another is to view either the pattern on the right, where these triangles are moving around, but they seem to be just moving randomly, and on the left, the triangles are dancing, hugging, and kissing, essentially like social interactions, and the question was to make that judgment. And a third of the 7 tasks was what we call working memory. Look at an image and say, is that the same or different from the one you saw two back? And we mapped these results onto the entire brain, the inflated maps of the type you've seen before, but now for the left and right hemisphere from the lateral and medial aspects. And we looked at the things down in the subcortical regions and on the cerebellum that you've heard about before that we flattened out. And what do we see? Well, some what I think are pretty fascinating stuff, but it's complicated. Let's walk quickly through it. So this is listening to stories versus a baseline of no particular task. And in yellow and red are the strongly activated regions. In green are the regions that are actually turned down or turned off by this. But the exciting part is the language-related areas on the lateral aspect, particularly in the temporal lobe, in the frontal lobe, and this little patch here that wasn't expected, but we found it consistently. And you can see there's stronger activation in the left hemisphere than the right hemisphere. That's expected from a lot of previous studies, but that there is a somewhat mirror image, slightly damped down in the right hemisphere, is interesting. And there's something going on in the cerebellum too, just for good notice. In a different task, looking at these triangles and saying, are they socially interacting or not, you see a totally different pattern. There's some overlap, but generally the pattern is very different in both the left and right hemisphere, and there are hotspots galore to be thinking about what they're involved in. The third and last task we take a quick look at is the working memory task. So when you're trying to remember what you just saw, what's different is mostly up in the frontal lobe and also up here in the parietal lobe. And so the general punchline is when your brain is doing something, like I hope you're still awake and listening to this talk, all sorts of stuff is going on. Different tasks engage networks that are complex in their spatial organization and partially overlapping. So it's not just that when you listen to a word, one little patch is turned on and that's it. So lots to think about. And more to think about when we move to our next exemplar modality, what we call resting state fMRI. So this is a few moments in the life of a volunteer who is in the scanner just looking at a blank screen, well actually it had a small crosshair to fixate, and the brain is very active in a complex symphony of activation and correlated signals. If we take that signal and look at the time course and see how does it fluctuate at one region and a nearby or distant region, is it similar in its time course or very different, correlated or anti-correlated or in between? We can do that for all possible combination and get a giant matrix of connection values or correlation values that we call a functional connectivity matrix, or a dense connectome is another lingo we use. We can probe or interrogate that entire matrix one row at a time. So if we take this location here and say, who's firing or signaling in a similar time course, we see strong correlations nearby, but at a distance in the same hemisphere and in the opposite hemisphere. And a different seed location, as we call it, shows a very different pattern. So all sorts of interesting correlations. And we use these different modalities or types of information to try to bring it all together and say, first of all, what does it tell us about how the human cortex is mapped or parcellated? And so as a reminder, this was the classic Brodmann map you've seen before. This is the classic Milo architecture map that said there's 4 times as many areas. Other studies using modern fMRI techniques have come up with other maps. Here's one from a colleague in Harvard. Here's one from Steve Peterson's lab at WashU. They've used modern methods to try to get better maps, and they are indeed better than they used to be, but they're not perfect and they hadn't agreed with one another. So Matt Glasser, whose name I mentioned before, worked with me and others in this Connectome project to get a better, what we call a multimodal approach that uses information about the structure, the topography, the connectivity, and the function. And we showed that we're able to parcellate not only a group average, but accurately in individual subjects, as you'll see. So I'm not going to walk you through the gory, but to me intriguing details of how we get this map. I'm just showing you the punchline, the bottom line, which is this colorful map that shows 180 distinct cortical areas in the left hemisphere and a matched set, remarkably, but not perfectly symmetric, in the right hemisphere. We can see these we can give them labels using names, many, almost half from the literature, so not all of these are newly discovered, but more than half of them actually were newly identified, sometimes by taking a previously published one and parsing it more fine-grained. As I said, we can do this not only in a group average, which tells us the kind of the big picture, what's consistent across many individuals, but Matt was able to train a fancy algorithm, what we call a classifier, aerial classifier algorithm, to recognize the signature or fingerprint of each of these 180 areas in each hemisphere in every individual by comparing it to the group average. And so here's the individual subject parcellation for one subject, here's another subject, and basically each area of these 180 is identifiable in nearly all individuals, and each of these areas vary in their size, shape, and position, not just like the pepperonis I showed you a while ago. So, we've made a lot of headway, and other studies that I don't have time to tell you about have tried to correlate the signals, the signature of functional connectivity that I described with things like behavioral characteristics, one notable one being intelligence, fluid intelligence. And there turn out to be interesting but complex and somewhat subtle correlations between brain connectivity and intelligence. And if you're curious, we can talk more about that afterwards. But now I want to move on to where we're at now, the so-called Lifespan Project to study development and aging hand in hand. But I'm going to give you some context for this by saying, why do we care about young brains and old brains now that we've gotten the progress I showed you before on the young adult humans? So brain disorders strike at all ages in very diverse ways. One of the first ways that I got involved in studying it is in an unusual, fascinating disorder called Williams syndrome, which is not just everything's gone wrong, but Williams syndrome children are very social, very loquacious, very good at some things, and very bad and awkward and inept at others. So it's some things stronger, some things not so great. And when we looked at just the folding pattern across individuals with the Williams syndrome compared to healthy controls, we basically found a whole bunch of differences in interesting regions, some of them involved in higher cognitive function, like I touched upon before. That's just one teaser example. Another has to do with preterm birth. So as many of you know, preterm birth is increasing in incidence, and it's all too great a risk, greater than 50%, for some kind of disorder that can turn out to be autism or attention deficit or language disorders. All sorts of things can go wrong. And part of the challenge or the issue is that preterm birth doesn't give brains that mature entirely perfectly in the intensive care unit. So I showed you a while ago that at healthy term birth, the newborn brain is nearly as convoluted in the newborn as it is in the adult. And these are just two examples as controls for our comparison. If you take infants who are born very prematurely, like 24 to 26 weeks, they can be kept alive. The ICU, neonatal intensive care units, are extremely good at keeping infants alive, but they're not so great at keeping the brains growing healthily. And so in many of these preterm infants, the cortex is not folded properly, particularly here in the temporal lobe in some individuals. So it appears that there's a selective vulnerability in some brain regions, and these are fascinating and obviously clinically extremely important to try to understand better and find better ways to keep brains growing properly. So that's just a couple of quick examples on the infant side. If we look at older adults, here's a type of scan that you've seen before of a healthy young adult. Here is an older adult where there's some atrophy, most notable because the ventricles are bigger. Here's a severely atrophied elderly person, and you can see how much gray matter has been lost through this atrophy. And we want to know, even though this individual is reasonably healthy, what's going on in the brain during healthy aging as well as during disease conditions such as Alzheimer's, that is a scourge upon our civilization nowadays. So magnetic resonance imaging and PET scanning are helping decipher this. So here's an example of a particular brain network that is turned off during tasks. It's called the task negative or default mode network, and it occupies several regions. And I mention it because that's a region that is burning energy more rapidly, elevated glycolysis, glucose consumption in the healthy brain. And in Alzheimer's disease, there's reduced glucose metabolism and deposition of one of the trigger substances that sought to provoke Alzheimer's. The so-called amyloid is not deposited uniformly, but in regions that are correlated with this specialized default mode network of the brain. So work on this and many other aspects is going on intensively around the world with hotspots of exciting discoveries going on at WashU. And so we need better neuroimaging tools and better information about what goes on both in healthy brain and disorders. One last example is clinical depression. I think many of you, some of you may have experienced it, and probably most of you have family or colleagues who've known that clinical depression is prevalent and doesn't always respond to drugs. Emotions and moods are managed and mediated by networks that I've touched upon before. This purplish region is one that's involved in mood control. And PET studies from years ago have shown that there are abnormalities in glucose sugar utilization in depression in this particular region. And new methods of stimulating the brain are offering some hope for alleviating the severe symptoms, but there's a long ways to go, again, for studying brain disorders. So, coming back to the Lifespan project that I mentioned, it builds on the success of the Young Adult Human Connectome Project that, as I mentioned, wrapped up a couple of years ago. WashU is one of 4 centers around the country that are involved in 2 projects. One of them is the Lifespan HCP Aging Project that started in 2016. It's targeting to study 1,200 older individuals from 36 up to centenarians. We've been acquiring data for more than a year. We'll start releasing data next year. The development Lifespan project started in the same time frame and is studying children and teenagers from 5 to 21 years, again scanned at 4 different institutions. There's another project that we're not involved with, but just for context, there's a baby connectome project that's studying children from 0 to 5 to cover the postnatal lifespan, and there's a prenatal and neonatal project in United Kingdom. So altogether, this collection of projects is going to give us an enormous amount of information and hopefully deep insights about what's going on in the brain in health and other projects studying brain disorders. For those of you who are potentially interested in volunteering for the WashU-centered Lifespan project, or if you have a relative or friend who's interested, we encourage you to sign on if you're in this age range or know somebody. And you can contact Tiara Redrick at this number or that email. I've set some flyers. There are a few up here, and there's a whole bunch outdoors, so take one on your way out if you might be interested in using that. Tell your friends, neighbors, and relatives. There is compensation for this volunteer donation of time. And if you have friends or relatives in Boston, Minneapolis, or LA, tell them about it too, please. So to wrap it up for the big picture, understanding the human brain, as I said at the beginning, is a grand challenge. The Human Connectome Project is helping elucidate brain connectivity and its variability in health. We've already made major discoveries, many more still to come. We're not going to get a miraculous complete connectome all the way down to the microscopic level. That's well outside current technology. So we have to manage our expectation and be optimistic but be critical about evaluating all this complex data. We're looking for volunteers for the development and aging lifespan projects. And future studies of diseases, development, and aging will aid, we anticipate, in not only understanding but better diagnosing and hopefully eventually treating these brain disorders. So to take a 20,000-foot view, we saw a Earth Atlas a while ago. Here's a classic vintage millennium or centuries-old Earth Atlas. Here's a century-old brain atlas. We moved to books to store lots of information about brains and the Earth. The miracles of satellite imagery transformed Earth mapping, MRI, and other sophisticated methodologies are transforming brain mapping. Google Earth and the sophisticated computations and visualizations it can do have utterly transformed everyone's lives. And we hope that the world of connectomics is going to transform our understanding of the brain. So all this owes a tremendous amount to hundreds of people over many years. I don't have time to single all of them out. I've mentioned a few: Matt Glasser for many contributions, Deanna Barch, the co-principal investigator Camille Ugerbill up in Minnesota, Steve Smith in Oxford, Dan Marcus, the informatics guru at Washington University, and that's the story. And we're right at 8 o'clock, so we've got plenty of time if you have questions. Question in front. How much information does the human brain contain? That's a great question. We don't have a complete answer for sure, but I think I mentioned the number, something like 100 or more trillion synapses. A synapse is a communication unit. It's also a storage unit. It has a strength that is related to how much information it has is able to manage. And it's not the same as a bit of information, but to first order, it's got to be on the order of one or a few bits of information it can store, and that rate of information it can communicate. So the total amount of information we have is nothing like petabytes, in my view. It's probably more in the many gigabytes, but not necessarily even terabytes. Other experts might disagree. I think it's actually it's a fun and valid question, even if we don't get hard numbers, to get into the right ballpark. The other thing is how much can it communicate, what's the bandwidth in megabytes or gigabytes per second. And since neurons communicate information rather slowly, a few bits per second, even if you take the 100 billion cells we have, that's not this matching the bandwidth of modern high-speed, high-performance computers. So the reason we like to think we're still smarter is not because we have more straight compute power, but because we have better, in a sense, smarter for real-world conditions. Computers and robots are getting better at managing real-world conditions, so this is a rapidly moving landscape. But one of the reasons, besides fascination with the brain and wanting to cure diseases, one of the reasons for studying the brain is to see if there are principles we can learn to help better design computers to make their, quote, artificial intelligence more real-world intelligent. And that's starting to happen as well. Question over there. A fine question. So I'm not a clinician, so take my answer with a grain of salt, and it's something I've read a little bit about. I'm a firm believer, just in a personal context, that exercise is good for the body and it's good for the brain. But how is it good for the brain? I think there are some perspectives and insights on that. I don't feel expert enough to really comment in detail, but there are, if you read about it, there are, maybe somebody else in the room knows a bit or two about it, but it's something to take seriously, that healthy bodies and exercise bodies do improve cognitive function through ways we would like to understand better. Way back? So I do have a comment that does not come from my specific experience with the Human Connectome Project, but just some general awareness. So years ago, back in the 80s and 90s, it was widely presumed that all neurons are born before birth and basically nothing else happened postnatally. Then it became clearly shown in animal models that there's a lot of so-called postnatal neurogenesis, even into adult mice and other species, and even in monkeys. Then there's more recently been evidence that even in human brains there is postnatal neurogenesis, new neurons in certain regions like the hippocampus involved in memory, the olfactory system involved in smell. And I didn't see that particular article, but I've heard by a kind of informal grapevine that there's a challenge to this the earlier evidence about whether there is human postnatal neurogenesis, but I don't think that battle has been resolved. I think there are 2 points of view, and time will tell which one holds more solidity. Yes? Yes. I do. I'm glad you asked. So it turns out that in these what we call lightly myelinated regions, which we can see very clearly in the whole brain imaging, painfully detailed anatomical studies in postmortem humans and in animal models have shown that these higher cognitive regions basically have neurons that are physically larger cell bodies, more and better dendrites, longer dendrites, more synaptic inputs. It's like they're part of a more sophisticated, fancier compute power computer chip compared to the dull old little sensory areas that do their job well, but they, by this hypothesizing fancifully, in the sensory areas, they can do just fine with small computer chips, more vanilla information processing, and cognition, we can guess, might benefit from these more elaborated neurons and synapses. But exactly beyond that, what are they doing that takes advantage of that, we don't know. The axons that come into these regions are still myelinated, so I don't think they're necessarily conducting slower. There may be fewer of them per unit of volume or area, but it may also be that on closer scrutiny, they may compute better, but not necessarily as fast. So there may be some trade-offs there. These are fascinating questions that are not answered but can be attacked with current technologies. Sorry, what brain? Oh, what nurture factors influence brain folding? We certainly don't know, but I would hearken back to what I showed you with preterm infants whose brains don't fold properly when they're in the NICU, and something about that environment is not good. It's a nurturing factor that either something is absent that should be there, or something that's present, like some of the medications they have, or some of the respiratory controls they have, are causing problems. So not only fascinating but hugely important, but it's tough to get a firm handle on that as yet, but very important question. How much change in folding is there from birth to adulthood? A reasonable amount. So when I said the cortex is nearly as folded at birth as it is as an adult, I also mentioned it's growing threefold in its total extent, and it's also getting somewhat more folded. And there's evidence that differences in individual folding patterns may relate to characteristics, intelligence, and other phenotypic characteristics. So these are important questions, and hopefully the advanced technologies that what we call the HCP style of getting high quality information and analyzing it very carefully will give us better answers to questions of the type you just described. There are also large-scale projects in addition to the Lifespan Project. An even larger one is called the ABCD Project, Adolescent Brain and Cognitive Development, that's studying 10,000 kids from 9 years to 19 years. And that will tell us about changes in those individuals followed over a decade that will help us inform better answers to that kind of excellent question. Sorry, do I feel that the pattern or activity of the brain is what? Consciousness is, I wouldn't say it's a hot topic, but there is actually legitimate scientific exploration of, is there a seat or center of consciousness, or what is consciousness, other than that we have it, those of us who are awake at the moment. And I'm a skeptic that we're yet hot on the trail of what's going on. And I have a colleague, Christoph Koch, who's hypothesized that a particular little structure called the claustrum, that's one of these subcortical structures, is somehow especially involved in consciousness. Well, ideas are cheap, and there might be something to it, but I'm not swayed strongly by the evidence I've seen before. And I have a hard time wrapping my head around it. I can think more about when I perceive your face, even though it's at a distance, I'm attending to that little tiny patch of my visual world. Now I'm attending to somebody else in the room. When I perceive something, we have a lot of information about how information goes from early to intermediate to very late stages of what we call visual processing. And we have examples of, even in living human subject, individual neurons that respond preferentially to one or a very small number of individual faces or entities. So we're getting handles on higher levels of perception. That's not the same as consciousness, and my ability to grapple intelligently with that kind of stops at thinking hard about perception. Well, no, I'm utterly fascinated. So when I was at Caltech, Roger Sperry got the Nobel Prize for being part of the studies of split brains that help point to this dramatic differences in the apparent function of the left versus the right hemisphere. And we see definitely language and other asymmetries in our maps of the human cerebral cortex. But as I touched upon, I'm more struck by the presence of a high degree of symmetry in the wiring and functional organization, and it kind of collides with this idea that there are profound differences between left and right brains. And so I don't discount the left brain, right brain hypothesis. I think it needs serious revisitation and reframing in the light of modern data, but not to throw it out entirely, but to say it's not anywhere near that simple that our two hemispheres are largely independent. The other thing about functional connectivity that I showed examples of is that the left and right hemispheres are talking very closely together in a coordinated way, and that is challenging for saying they're just two different computational systems. Yes? Or can I give an example of how? We're still challenging. It's a big challenge to synthesize, even though we can take information about these different modalities, the types of information, we can use them side by side, but how they coordinate and how a particular pattern of connections gives rise to a particular task, ability to move or think in a coordinated way, we're still scratching the surface. For those of you who are young and looking for something fascinating to do, we're not tapped out. There's a ton more to be learned, and I would encourage anyone who's fascinated by this to try to drill deeper and help us solve these great mysteries. If I haven't answered it satisfactorily, come up to me afterwards and we can pursue it. There's a couple of papers published. I don't remember the authors, but some papers from a Norwegian group, among others, who have reported these correlations, that the folding patterns correlate with IQ. Now, that doesn't mean they obviously cause it. What's underlying that correlation is still a mystery, but there's something to sink your teeth in. So if you want to learn more, I would suggest if you just Google folding and IQ, I bet you'll get a hit or two that ties into that literature. Yes, male brains and female brains are different. Female brains are leaner and meaner in the sense that they are literally about 10% smaller. But that doesn't mean they're less capable. It means maybe they're not wasting so much space on too much myelin or something like that. So from the Human Connectome Project, there have been some studies that have explored differences related to sex, and I'm not, because there are hundreds of those publications, I don't have my fingertip mental access to everything that's been reported on that. There are some intriguing things that are emerging, that it's not just women are not as good or better, but differences in the functional networks that carry out certain kinds of processing or tasks are starting to emerge, and some of these may pass scrutiny, others may fall by the wayside, but it's great to have these very rich, high-quality, consistently acquired data sets to explore these things. And in the Lifespan Project, one of the things we're doing for the children is concentrating extra scan time and participant numbers on the peripubertal range, where a lot of brain changes occur in both boys and girls. Can we get a better handle on that? And in women in their mid-50s, we're looking at the perimenopausal range, where there are reports of menopause is impacting memory performance, and it'd be great if we get a better handle on that. So these are very much accessible and important questions. I wish I knew, so I'll hasten to say it for the start, that I'm not familiar with that literature, and there's reason to be hopeful that some of these therapies and approaches to brain improvement will be beneficial and will stand the test of time. Some of them are probably going to fall by the wayside. And there are so many ideas and theories out there. I happen to have a particular not only fascination with, but a family interest in autism because I have a grandchild with autism. So I've been reading about that. The history about how scientists thought about the brain and in the context of autism has changed so dramatically that we can look back on even a decade or two ago, things that had traction are very reason to be very doubtful about now. On the other hand, there are therapies that are, I think, probably very important and likely to at least benefit to some degree. So we need better research, carefully executed research at all levels, from the design of behavioral paradigms to careful control of everything up to and including if there's scanning involved, getting the best quality data and analyzing it carefully. So these are expensive, they're not easy to do, but that's the right way to get a better handle on a whole host of fundamentally important questions about the brain. Yes? The short answer is not yet, and that's kind of a definitely not yet. The predictive capabilities of, even with the improvements that have been made with the Human Connectome Project, show that there's still serious limits to how much we can infer about an individual's capability or life experience from, at the level of mapping one individual. I mean, I mentioned one of the measures that has yielded some traction is predicting IQ based on brain scans and functional connectivity. But that correlation, that predictive capability is not so great. So there's been another approach, which is to combine a whole lot of measures into a single, what's called a positive-negative axis, where things that are good to have, like high IQ, good working memory, good life satisfaction, versus poor sleep, aggressive behavior, and all that. That combination is more predictive at the level of individual subjects. So there is some forward progress, but the miracle cure or hope that we could just dive into this data sets and, like you say, that person's an engineer, that has not panned out, and probably best not to be too over-optimistic about what reality imposes on this approach to science. Yes? So, not in these projects I've been involved in, by intention, and the funding agencies, National Institutes of Health, said basically study healthy brains and get ourselves a strong baseline of what's in the normal range, but not a supernormal. We have taken subjects who have, they have been to doctors for various things, but they're generally considered in the healthy range. There's another set of projects that are still ongoing, so it's early to talk too much more about them than tantalizing. They're taking these advances in the methods that we developed with the HCP and putting them over into disease conditions. And there are 14 different projects. Most of them are studying mental disorders, psychiatric disorders, but dementia, vision loss. I don't remember one relating to what the condition neglect that you described. But if these other projects yield pay dirt, there will be added interest in exploring a wider range of abnormalities and pathologies. that's work yet to be done. Yes? Do I think that in human evolution? Well, it's in evolutionary context, the human brain has evolved very rapidly relative to many other species where we have a handle or prediction. So, but the changes in the human brain over the millennia that we've acquired strong culture and strong language capabilities, it's not so clear that there have been major changes in, like the folding pattern, that underlie that. That's probably more of a nuanced reorganization of circuits that were evolved under one set of pressures to, manage the environment, and just suddenly became capable of mediating the conversation we're having this moment. If you think, can a pair of chimps have a conversation like this? No way. They have only the crudest, rudest type of language capabilities. But what is it that's changing in the brain in recent history is still pretty much a mystery. But at another level, why do we have so many more folds and so many more neurons than a chimpanzee does? That's controlled basically by factors in early development where the cells that divide and proliferate to become subcortical structures amplify themselves to some degree, but those that are going to become the cerebral cortex, they just make many more cells, undergo many more cell divisions, and lo and behold, we have a bigger brain that's dominated by the cerebral cortex. So there are, you can think intelligently to some degree about these issues, but there are a lot of fascinating unanswered questions for sure. Yes? Yes. Well, the honest answer is we did not overcome the problem of resolution. The multiple macro, micro, meso connectome that I illustrated shows the scope of the problem. What we were able to do was to improve the spatial resolution by factors of 2 or 4, which is a lot, but when you're talking about the difference between a 1 millimeter voxel and 500,000 neurons, that shows you the scope of the problem. So that's part of the reason why, as cool and fascinating and informative as in vivo neuroimaging is, one of the reasons it doesn't do so well at predicting whether you're an engineer or an artist or whatever is because we don't have enough information about the detailed circuitry, and we can look around the curve and say, is there going to be some miracle new method? I believe they'll get better incrementally and with intense effort, but I don't expect, even in a fanciful future or a century or a millennium from now, if our species is smart enough to survive, which is questionable in its own right, will the technology have taken us all the way to understanding the full human connectome? I guess I'm honestly a skeptic on that front. There are limits imposed by the laws of physics. Yes? It's a question that fascinates me. It even bugs me because I've seen more papers published that claim to disprove that hypothesis than that support it. So you could call me that I've got my head in the sand, but I'm a skeptic who looks hard at the evidence, and the publications that claim to discount it are flawed in ways that I could, and I could go on, but I won't. But there are problems with the data in critical studies. But what I would dearly love to see is a convincing proof, and that requires showing not only that neurons can generate tension, which they do if you put them in a dish, but that they do generate tension in the living brain in a way that my model explains it. And I can think of ways to use the Human Connectome Project data and the patterns of connections and the patterns of folding across many individuals to inform that debate, to support it or rule it out. But that would be a huge amount of work that we're not ready yet geared up to do, and it would not be compelling. So some questions, I mean, I would pay $10,000 if you gave me a sealed envelope and it said, here's the answer. But I don't believe that the current technology is yet ready to incisively resolve that question right now. Well, there is a technology called elastography that actually measures how elastic a tissue is. Its resolution in space is so coarse that it doesn't tap into this. If that could be somehow miraculously zoomed in so that it could measure the elasticity at the level of a millimeter or so, I could think of very significant questions that it could compare the elasticity of cerebral cortex versus white matter versus cerebellum that would give strong clues as to whether that hypothesis is supported or negated. So it would be more than an incremental advance, wouldn't necessarily prove it, but proof in science, as you probably know, is more a matter of strengthening or weakening the evidence rather than saying we solved it to all possible further debates. Over there? I mean, functional MRIs? I'm not sure how one would do that exact experiment. There are experiments in which, in two different scanners right nearby, two individuals are scanned at the same time, and there's efforts to use the fMRI data to show. I don't know quite how those experiments are done because I've never read them closely, but they apparently have some cool findings with regard to coordination of fMRI signals in people in the same scanner at the same time. So there are some clever techniques. There are also a variety of techniques that decode the brain, going back to some of these other issues. If you listen to the brain in real time and just say, the person is looking at a movie or looking at a string of letters or some pictures, can you predict from their brain activity what they're looking at? And the answer is yes, you can to an interesting degree, but it's still not to a degree that somebody listening to my brain could recapitulate who I'm talking to and what I just heard. That's far beyond the current scope. One. Okay? Yeah. We certainly, not that I'm aware of from current studies. We could imagine, a serious question from Connectome or HCP style data saying, are there correlations with mathematical ability, for instance, that are diagnostic across individuals? It goes back to, is this person an engineer? Is this person good at math? We'd dearly love to be able to probe at that level. I'm not sure the sensitivity is there yet. It might get much closer. And then you could say, not just are women more mathematically inclined than men, but you could probe a profile. Women are not just one lump group. They're amazingly diverse, and so are men. And so understanding more, can we tap into what talents individuals have, whether they're male or female, and understand better, or perhaps give better guidance as to what they might want to do with their lives. Those are interesting and not so loaded questions, I think. Thank you for your attention and stamina. Oh, and don't forget to pick up flyers if you're at all interested.